The microbiome. You've probably heard about it on NPR or read about it online. It may not be a subject dear to your heart, but this collection of microorganisms, from bacteria to fungi and viruses, is found in your mouth, on your skin, and in your gut. For me, what is amazing is not only that we have maybe 10 times more bacterial cells than human cells in our bodies, but also that a group at Oak Ridge National Laboratory is studying the microbiome. It's pronounced microbiome, not microbiome, which might be something you think Oak Ridge would be doing. So I found this out by reading an O&L news release on the most recent researchers to be named corporate fellows. One of them is Robert Hettich, who taught a class I took years ago at the Oak Ridge Institute for Continued Learning. I retired 11 years ago. It turns out that I learned from the latest news release that he has been studying the microbiome by using advanced biological mass spectrometry. So today he will tell us how the microbes in the human gut play roles in health and disease. Bob is a UT Battelle Corporate Fellow and leader of the Bioanalytical Mass Spectrometry Group in the Biosciences Division at Oak Ridge National Laboratory. Bob and his research group at ORNL helped pioneer the field of metaproteomics, which is focused on using advanced mass spectrometry to measure the suite of microbial proteins in a variety of human microsome systems. Before joining the ORNL research staff in 1986, Bob, a native of South Dakota, received a doctorate in analytical chemistry from Purdue University. He is a joint faculty member with the University of Tennessee's Microbiology Department and with the Bredesen Center for Interdisciplinary Research, where he was named Outstanding Faculty Member in 2023. So please join me in welcoming Dr. Bob Hettich.
All right, can you hear me well enough? That's pretty good. Okay, let's see here. I don't need a phone call right now. All right, well, it's good to be here. Good to have the opportunity to speak to you. Wow, I sound really loud here and stuff, so you can hear me as well as you can. Okay, I should speak loud, right? Okay, so it's really, I know many of you from work experience. I've just passed year 38 at the lab, and I'm kind of looking at 40 now if I can survive. We'll see how that plays out and stuff. What I want to do is I want to talk to you a little bit about the research we've been doing in the microbiome. And it's a very interesting concept. I'll tell you, it's fueled a little bit by my interest as a father. I have a 40-year-old daughter that has Crohn's disease very badly. And I've seen this from a father's standpoint, including multiple surgeries and all the stuff that goes with it. So it's kind of passionate to my heart, the aspect of microbiome related to inflammatory bowel disease. And I'll talk about that a little bit today. I do want to point out that, one thing that I want to convey today, and every time I give a version of this talk, I find there's a lot of response back. So if I can work it well, I'm going to try to shorten my talk a little bit and allow a little bit extra time that we can just visit about stuff. If that's fair, right? So I think this is really playing out into a new way to think about health and disease. And I'm going to try to give you some concepts as we kind of go along, in my opinion, about what this looks like. I'm at Oak Ridge National Lab, and we want to do something like we want to take a gut microbiome, we run it through a shiny mass spectrometer and come up with information on the other end, right? So that's kind of the general goal. Now, knowing this is a pretty mixed crowd, I want to divide my talk into three parts. I first want to tell you, what are microbiomes? What are they and why would you care, and what do they kind of look like, and what are some of the science questions? And then I want to talk about it, how would we measure them? Very complex samples, and if you know a little bit about my background, mass spectrometry is a lot of what I've done for over 40 years. So we're going to use mass spectrometry to try to address questions. Then I'm going to give you a case study of preterm human infants. I'm going to show you what that looks like in the context of what kind of things we would learn. So let me start off from a general background just to kind of make sure we're all in the same place. You may not be aware that around us in the world is a whole unseen universe that most of us never realize. And if you don't think about it, you don't realize there's billions and billions and trillions of bacteria on this planet in every environment you can possibly think about. And these exist in little communities. And so growing a single isolated lab bench culture is very artificial. Bacteria exist in mud puddles, oceans, and a whole range of stuff. In every place they exist, they're always composite mixtures of things. And most people will recognize bacteria. There's certainly other microorganisms. There are archaea, there's viruses, there's microeukaryotes. They all live in systems, and many of them will never be brought back to the lab and cultivated. They exist in an environment that's hard to replicate inside a lab. So if we're going to measure them, we've got to think about technologies that will measure them endogenous where they're at. We bring a sample in, we're not going to be able to cultivate them. We got to do something with a soil sample or a fecal sample in situ. A lot of these are part of the human body as well. And so I want you to, I don't know how well you can see, if you can look at the middle picture, I want you to look at that for just a second. This is a cartoon diagram, and I want to point out a couple of things, that these microbiota, these small microorganisms live in there. You see how they're spatially segregated. There's some order to that. There's certain members that only exist close to other members, and they're never with other ones. So there's some competition. We'll talk about that in a little bit. There's some temporal aspects. So the bacteria that live in your small intestine are very different than the colon. So there's a whole range of stuff. Most of the time, you never see microbiota, right? And occasionally there's these microbial mats you see way on the right-hand side that have these beautiful plumes that are colored, and they're actually macroscopic evidence of biofilms or other stuff. So these microbial communities exist everywhere. It's only recently that we come to realize the importance that they play in oceans, in environmental control, in ecology, whatever, and in human health. And that's the source of what I really wanted to look at. So if you think about microbial communities, there's a koi pond picture on the right-hand side. If you look at a very complex system and you just look at the picture on the right-hand side, you see there's plants, there's animals, there's algae, there's a whole bunch of stuff that you don't see all existing in some relationship that's fairly complicated. And so we can start to ask questions about these microbiomes, these microbial communities. How did these, oops, let me go back here. How did they start in the first place? How did they assemble? Who gets there first? Who sets up shop first? Usually there's an initial colonizer that then recruits other bacterial members to increase complexity. What happens when a community ages? What happens in a microbiome in your gut when you're age 20 and now you're age 80? What does that look like? Is that stable? Do the members change? A lot of open questions. But real important to me is the middle part about how do communities function? So these things are like little townships. So these communities exist in very specific patterns, and they talk to each other. They exchange metabolites and proteins. They keep track of populations. You're okay as long as your family doesn't get too populated. And if you do, I'll kill part of them. And so this is called quorum sensing. There's a whole regulation of control. And so these communities, they basically cooperate and compete for resources, much like humans do, right? And so when there's nutrients present and when nutrients start to dry up, the 500-pound gorillas will take over and they'll dominate, and that causes problems. The microbiomes also protect each other. And so these communities that live together, and an invading bacteria comes in, they'll kill it because that's foreign to the community. So there's a whole elaborate set of balances that are really of interest to me, and I'm going to try to bring out some science questions in a minute. The last one is, how did they interact with their host? The bacteria that persist in your gut will persist for over decades in a very stable aspect. How does that happen? How does the human system, how do the machine guns on your epithelial cells not kill them all off? So there's got to be some synergistic balance about, you're allowed to live in my gut as long as beneficial things happen, right? And so if they don't, sometimes the trajectories go toward disease. We'll talk about that in a little bit. So this is a wonderful time to do chemistry. And I got to tell you, my hat is analytical chemistry. So I'm a hardcore mass spec guy from Purdue, learned a lot of mass spectrometry, came to Oak Ridge to do that, and realized biology is the future. And I really want to do biology with mass spec. So it's interesting, my position at UT is not in the chemistry department, it's in microbiology by choice, because that's the people I want to talk to. So there's a whole family of technologies. This is called the omics toolbox, and you can see it on the middle side. And so there's a whole way of getting samples out. And if you look at the left-hand side, we can do this ribosomal RNA, counting basically. What are the bacteria that are there? And then we can dive all the way into the genes, the proteins, the RNA, and the metabolites that are evidence of things going on. And they're all called, I'm going to talk to you a bit about metaproteomics, is a particular technique of measuring the proteins. But you see they're all connected by arrows together. And so in some cases, the metagenome will tell potential function, what can it do? But the community may be doing something a bit different. And I'll give you a couple of examples of that. So I tell my graduate students, what a wonderful time to do science. I would have killed in the early 90s to have this type of capability. And I have new graduate students walk in and like on day one, they're doing measurements that I wouldn't even thought about maybe 10 or 15 years ago. So what a great time to do science. So we looked at a variety of these communities over time, and this was an analytical chemistry article we published a while back about what is this technique called metaproteomics, and I want to kind of define that. It's the ability to basically look at all of the proteins in a community, all of the proteins that would come from one fecal cell, and be able to measure what's happening. And this started for us with a collaboration with UC Berkeley. And if you look at the picture on the top left, this is a mine in Northern California that's leaching sulfide out of the rock. And the water pouring out of the mine is 1 molar in iron. It's 40 degrees centigrade and it's pH 0.5. So this is like hot sulfuric acid. And on the surface of the water is pink snot. Do you see it here? And the pink snot is a microbial biofilm that's basically not only living, it's surviving and thriving on boiling sulfuric acid, essentially. And so this is really like, how in the world, what in the world is going on? How can these extremophiles exist on this? And it's a very interesting, and that sort of launched the whole approach of trying to think about that. You can certainly think about, microbiomes that live in plant roots or tree roots or endophytes. There's a whole series of things that could be of interest. Okay. So, So let me lead you through a little bit about, okay, if we're going to do measurements on this, we have to translate my lab. How are we going to do a measurement to get information to answer a question? And I want to briefly take you through 3 things. First of all, how do we get the samples out? If we have a soil sample or a fecal sample, how do we get microbial proteins out of fecal material? Not completely easy, right? How do we make it compatible with mass spectrometry? And then the middle section are the measurements that we're going to make. And I'm going to show you briefly what it looks like. We use a lot of liquid chromatography, mass spectrometry, and we think a lot about these figures of merit in the middle. How sure are we that we're right? How sure are we that we're measuring the right proteins and peptides and consideration? And then the bottom part, there's a tremendous amount of computational horsepower that's needed to deal with the data. This comes pouring out in a dramatic fashion, and there's a whole bunch of stuff. I won't go into detail unless you're interested about how to process the data. So I graduated 2 PhDs from my lab that never went in the lab. They never touched the mass spectrometer, and their entire PhDs were focused on how do we deal with the data. This gets very complicated and was a source of multiple research. So if we think about taking a sample for a microbiome, so hopefully I've kind of convinced you that microbiomes are important. If you think about taking a sample for a microbiome, I want you to look at the picture on the right-hand side for a little bit and just look at some of the dynamics that would be involved in us thinking about a sample. Give you a few seconds to look at that. It's a very common picture, right? It's a pond, but if you have to step back a little bit, do you see the multiple kingdoms that are present? Do you see plant, fungi? You don't see billions of bacteria that are in the water. You see that there's some spatial differences, right? In the water is different on the shore. So that sample would look different from a microbiome, right? What you may not also appreciate, there's temporal differences. If I went to this place in the spring or the fall and the winter, that same pond is going to look very different. And so the question come up, does the microbiome change dramatically as a function of those? How dynamic or how static is the microbiome? So we've got to think about how much water do you have to give me? What does it look like with all of the interferences that are going to cause some problems in my mass spec measurement? So if we go into the mass spectrometry, these are going to be very complex samples, and I'll show you in a minute. We're going to do a variety of chromatography methods, and that's the top bullet. We're going to try to separate the very complex mixture apart and then present it to the mass spectrometer. And this is going to all be done online. We've done a lot of analytical chemistry to figure this out, and my group is one of about 3 in the world that can do these kind of measurements. So this is what the process looks like. I'm just going to give you a flow chart a little bit. So we're going to start in the upper left. We're going to take a sample. It can be soil, it can be fecal material, it can be a variety of things. And we're going to then extract the media away from the proteins, and we're going to get the microbial proteins out. And then we're going to do something very stupid analytical. We're going to take 1,000 proteins, and we're going to digest them and break them into parts. So we're going to make 10,000 peptides. So one protein made a whole bunch of little peptides, and we're going to measure the peptides, but that's actually a better measurement to make. So we're going to do that. Now we're going to come into an LC in the top middle. This is liquid chromatography, and we're going to separate the peptides, present them to a beautiful mass spectrometer, and then we're going to measure the mass spectra on the bottom right. And let me just go ahead one side. I want to show you what that looks like. So the chromatograms on the top, we're separating over 240 minutes. If you look at the x-axis, for 4 hours, we're going to be pulling this very complex mixture part. It's probably got 100,000 peptides in. The mass spectrometer is scanning more than 5 times per second during those 4 hours. So we're just ripping along, measuring all of the peptides. And so if you see that green cursor there, if I look at that place, these are all the peptides that are eluting at that point in the chromatogram. What I want you to look at is, do you see the mass accuracy? Do you see 4 places after the decimal point? That's phenomenal. And so if we had a peptide at mass to charge 601.45, that's a mile away. And so this is the power of high performance mass spectrometry really being definitive. Now we're going to do something, if you don't know much about the detail, we're going to take that particular item and we're going to do tandem mass spectrometry. So inside the mass spectrometer, we're going to tell the instrument, take that mass to charge and accelerate it and slam it into a target gas. And when that hits, the ion falls apart. And it falls apart very specific. It falls apart not random, but by chemistry. It breaks the bonds between the amide, and you can see the pattern that comes out from this particular peptide. These are all of the fragment pieces. And what's kind of cool is when it fragments between the amide bonds. So that means the spacing between these are amino acids. You see that? You see the ladder? And so you can read this off. You can give a table and you can say that was serine, that's glycine, that's asparagine, okay, alanine. You can read these all off. And so you can do the sequence tags on that. So when graduate students join my group, I torture them. I make them do this by hand a few times, right? So I say, if you take this one and look at this one, can you find the amino acid peptide out of that? And they can do that with a little bit of work. I mean, I've done thousands of these in my career. What I don't tell them is we're making 2 million of these measurements per day. Okay, so then they go run screaming from the room. Okay, we go back. But the bottom line is, after make them do a few, we're never going to do it by hand again. And so the bottom part, we're going to match the measurement against the genome. We're going to do a pattern matching. And we're going to say everything that I measured is predicted in the genome, so I can tell what's right and wrong by filtering. And I'm going to come out with an Excel table that's about 20,000 lines long. Now, this is really cool because it's got a bunch of information about abundance, but here's the really cool part. You may not be able to read it. These are the protein names. This is reductive dehalogenase from dihalocoides. That's spectacular, and I would not have believed that when I first started doing that. The specific information that comes out of the peptides can be matched to a specific protein and an organism, and this thing will go basically 20,000 lines long. So you can start to say, if you look at abundance here, this reductive dehalogenase is way more abundant than this hydrogenase, for example, right? The mass spectrometers keep track of all of those details. So what does that matter? Now we have this detailed inventory, thousands of things, thousands of components can be sorted out. And I'm going to show you in a minute why that's important, why we care. The problem is, only to a mass spec nerdy guy like me would 20,000 line Excel sheet mean anything. To most microbiologists, that I don't know what to do with that information. And so what they want to get to is something that looks really spooky, and that's this. Okay. So don't get scared. Don't run from the room. You got to hang on for a little bit here. So this is called a KEGG map, and this is sort of the wiring diagram of the cell. And if you've not seen these before, it's kind of an encyclopedia of genes. And so without a lot of detail, there's patterns here that tell something about functional information. So this is fatty acid synthesis. This is the TCA cycle. There's nucleotide metabolism. And what I've colored in red are all the proteins that we measured that are mapped onto pathways. And a microbiologist can look at this and can start to ask questions. Is this organism happy or stressed? You can start to read these from the sheets, right? Then if I monitored it under different conditions or different times, the colors change, right? This may disappear and this part may come in. So you can see some of the details that we can get at a very fine level.
This is one organism?
This is one organism, but you can do it on multiple ones and you can see how it goes together and stuff, right? So this is kind of called the wiring diagram of the cell. It's kind of telling you the functionality that is done under certain conditions, right? So this is what it looks like in my lab. This is a postdoc, Wiley Zong, who left my group a couple of years ago. Here's the LC column. It's a really tiny piece of fused silica. This is the electro spray in the front of the mass spectrometer. And so we've got 6 instruments. We run probably about 360 days per year. They run 24/7 through the weekends and stuff. They're all automated samplers because they're really detailed measurements that are really pretty specific. Okay, so let me pause for just a minute. Hopefully you're still with me here. So what I've told you so far about microbiomes are incredibly important. They're all over the place. We should care for many reasons. Secondly, I've told you a little bit about how we make the measurements, mass spectrometry, they're complex samples. If we do liquid chromatography and the mass spec, we can start to sort out these very complicated mixtures. Okay, now at this point you'd say, I get bored here. Why do I care? Okay, so let's talk about the human microbiome then. And in order to do this type of research, we have to do the type of measurements we just said. So for most people, you're kind of aware that there's a microbiome. You read that in the popular news, right? And I find when I give versions of the talk, people normally say, when I think of bacteria, I think they're bad, right? Bacteria, I want to go to the doctor and get antibiotics and kill them off. Please don't do that. Okay, you would be completely dead if there were bacteria on your skin, in your mouth, in your gut, whatever. So the human system is very interesting. There's a tube inside our body that's open on the top and the bottom, right? And that's a weird condition that, it's open to the environment. And so in your gut, there are billions and billions of bacteria. And we'll talk a little bit more about what they are. There's a tremendous number of strains. They're highly diverse. They have very complex networks of how they work together. And there's things you can do to screw them up, right? There's things that you can do to perturb your microbiome in ways that you don't want to happen. And I think what's coming, I'm a little foreshadowing, I think all of us, everybody done eating lunch? I'm going to get disgusting for just a minute. Okay. I think my prediction is within 5 years, you'll give a fecal sample to your doctor and your doctor will sequence your microbiome and you will have a folder that contains the bacteria numbers and population that you have. And that's going to be important because even some of the drugs you take are dependent on your microbiome. So I think that it's exciting and I think it's coming pretty fast. So this microbiome is sometimes called a forgotten organ. And so Carolyn alluded to the fact that there's probably 3 to 10 times as many bacteria cells on your body as there are human cells. And this is an incredible, not an insignificant thing. And so the bacteria, you can see some of the things it does in the upper right-hand portion there. It actually helps with nutrients. Your microbiome actually attacks pathogens. And it trains your immune system. The microbiome actually trains your T cells to, have a function. And so if we think of the human genome, this was a Herculean feat in 2001, right? This is biology equivalent to landing a man on the moon, sequencing the human genome. And what was very disappointing is to find out that we're about the complexity of a fruit fly, right? So humans only have about 20 to 30,000 proteins, and it looks like worms and other stuff are about that level. Now there's modification. But the microbiome contributes at least 1 million more genes to your system in addition to that. This is not an insignificant player in who you and I are, right? And so this very complicated dynamic. If you look at the top left, you can see there's relatively few bacteria in your stomach because the pH is too low, right? But when you start to get into the gut and you get into the colon, there's something like 10 to the 12th colony forming units per mil. Fecal material can be almost 50% by mass bacteria. And there's an incredible load of cell. Why are they in there? What are they all doing? Are they good or bad? That's really kind of the concept that's come out recently. So if you look at a little bit of detail, in most of us, here's the epithelial cells in your gut, and they're covered by a mucus layer. And then there's bacteria on the outside, right? And my concept of bacteria sort of hanging on the side, waving in the breeze when nutrients flow by, right? And stuff. And in most cases, these are very carefully balanced. The green ones are the good ones. The red ones are the pathogens, right? But in some cases, something goes wrong in a way that we don't totally understand. And the pathogens, the red, take over by population or by function, and somebody's doing something they shouldn't be doing, and the scale tips. And this thing has been implicated in a whole bunch of things. And it's really interesting that cancer has a huge relationship to the microbiome right now in ways that are not clear. Autoimmune diseases, even Parkinson's, the gut-brain axis is thought to be something related in large part to the gut microbiome. So what is this? What's happening? What's going on here? One thing that happens when this situation occurs, this mucus layer gets thin thin. Do you see how this gotten quite a bit thinned? And thinning of the mucus layer is not good because that leads to inflammation and all sorts of stuff, bad stuff starts happening in your gut. So the question comes out, is the microbiome causing the problem or responding to it? Is it a cause or a correlation? And this is where microbial systems ecology starts to come in. Okay, how do we dissect that level of information? What's the level of detail there? Okay, so another picture. Here's on the right-hand side is the epithelial cells. On the left-hand side is the microbiome. And there's this really interesting interface in the middle. The microbiome talks to the human system by trading a bunch of metabolites and proteins. And those are things we can measure, right? And so in the red box is where my research lives. I really want to measure a lot of the metabolites and the genes that are indicative of either healthy or a disease condition, right? And so I talked a little bit about the complexity of what we're going to measure. So this has been implicated in a variety of things. Carolyn and I were talking even before this about the implication. If you look in the top right-hand side, in Crohn's disease, there's several bacteria that are really markers of bad things, right? And so the family, the genus of Bacteroides, but there's particular species that are elevated in Crohn's disease relative to healthy. A huge impact that most of us in the US are real concerned, obesity, right? In obesity, you want a lot of Bacteroides, and in lean people that have a high amount of Bacteroides, obese people have a lower amount. So there's some very characteristic signatures that are coming in. If we understood this, where are we going? You could go, get a probiotic and you say, I want to increase this level of stuff, right? So there's a whole bunch of connections, and you can read some of the, disease cases that are kind of related here. Okay, let me finish up the last part here with kind of a case study, and we've done a variety of things on preterm human infants. And so one of the interests I have, and I kind of interacted with a guy in Pittsburgh who's a pediatric surgeon, most babies are born with no bacteria in their gut, even though the mother has a tremendous amount of bacteria in the womb. They're sterile, basically, right? So from birth to about 1 week, all of a sudden here comes this onslaught of bacteria that this little baby has never seen before, right? The nurses that are touching them, the blankets, the pacifiers, the feeding types or whatever and stuff like that. And so this is kind of the pendulum swinging all over the place. What happens, the first bacteria there are normally facultative anaerobes, and they get outcompeted by somebody else, and it sort of rapidly evolves. And by about 1 year or so, or even less, that microbiome is stable now for the rest of life. And so you can imagine that depending on how the baby's born, if it's a cesarean section or a vaginal birth, that microbiome looks a bit different. Okay, let's do a test question for a moment. If a baby is born by vaginal birth, what do you think the microbiome looks like in the baby initially? The mother's fecal material. The mother's fecal material is what babies imagine. What does it look like when baby's born cesarean section? The mother's skin. So the mother's skin versus the mother fecal, that trajectory changes depending on mode. Whether they're breastfed or formula fed can also change it. So there's a lot of stuff that you can do to swing the pendulum a little bit. What's frightening though, and this doctor taught me, there's something called necrotizing enterocolitis. That's a horrible disease for preterm babies. This is when the guts perforate and the infants will normally die from it, right? And this happens primarily in preterms, not full terms, because of this wild pendulum swinging around. There's something that's correlated with that. We wanted to look in a little bit more detail at that. So we had 16 preterm samples, and you can see they're about half male and female. Most of them are C-section. They're fairly small. Most of these you can hold the babies easily in one hand. And we had 10 healthy ones and 6 babies that had neck. And so we thought this is an interesting kind of concept. Look at the far hand side. This ought to frighten you. Every one of them had antibiotics. Why would you give antibiotics to a healthy baby? That's because that's what doctors do for preterm. I'll come back to that in a minute. Okay, so this is complicated. Let me see if I can do this without confusing you too much. These are 4 individual babies. You can see names at the top, they're coded. Baby 3 is a healthy baby. Baby 19 and 21 are a twin pair. And baby 23 is a healthy baby. So 19, 21, and 23, those babies are all born at the same time, same hospital, same nurses are touching them and holding them. Baby 23 is healthy. Baby 19 developed an infection, got antibiotics. Baby 21 developed necrotizing enterocolitis and died at day 31. So this was an interesting set to take a look at what's going on in these systems. So what I'm showing you on the x-axis is time series of life. These babies are born, you know, a day of life 0 and a day of life 13 to 30, we taking samples. Then the baby died at 31. These other ones go out to 86 or 56 days or whatever and stuff. The colored bars are microbial taxa, and it's way too small for you to read what they are. I just want you to look at the colors for a minute. Do you see how all babies start out fairly simple? They're relatively low complexity early on. And as they progress, especially in this one, you see how complexity increases, very few types of bacteria. And with time, it becomes much more rich microbiome, which is part of the thing of, you know, the details. You can see there's a whole bunch of other stuff in there. But what is the other thing that you notice right off the bat? Let me pause for just a second. When you look at the colors, what do you notice?
Getting more varied in time on the good one.
Okay, they're all different, right? Okay, one unexpected thing, baby 19 and 21 is a twin pair, monozygotic twin pair from the same mother. And if you look closely, there's almost no colors in common. This was unprecedented. Nobody expected this. In the same hospital, this is the wild west of the bacteria that are colonizing a monozygotic twin can be completely different. And so, you know, this brought up a really important thing about it. Be careful about head counting. Let me give an example. So if we would, you know, somebody walked by the door right now and they looked in the door, they would count 30 people or whatever and stuff, right? If you walked by next week and looked in the door, you would count a different set of people. And you'd say, wow, those are really different conditions, but it may actually be the same meeting, just on different days, right? So you got to be careful about, you know, looking at who's there. What's more important is what they're doing, what the functions are, right? Okay, so we looked at this quite a bit about, is there any evidence, is this baby that died of Nick, is there something going on in the taxa? And it wasn't obvious from this particular viewpoint. So we backed up then to think a little bit about how does the human host, how does the human baby control the microbiome? What does that balance look like? And this is a study we published a couple of years ago with a postdoc in my group. So how stable are these communities during development? You know, going from 0 to 60 miles an hour in a month or so, what does that look like? How did these microbes respond to perturbations by the human host, for example? So we have 17 infants, and let me give you a second to look at that. On the x-axis is the gestational age, right? These are all preterms. They're born at about 24 to 26 weeks relative to 32. And then there's different sampling points. Every dot is a sampling point. Still coming in okay. And so, you know, remember, these babies are in the hospital not because of our study. They're in the hospital because of other reasons. So we would love the time points to all exactly match, but they don't exactly. Some of the babies are sick and they get taken off of the sampling, whatever and stuff. So we have 91 samples. If you count all of the points with replicates, we have 91 samples from these babies. We want to look at what does the microbiome look under these conditions. So the first question we ask is, what does the human host look like during these conditions? And so remember, in the fecal material, we can measure human proteins and bacterial proteins, and our informatics will completely point them to not confuse them. We know which proteins are human, we know which ones are microbiota in a fecal sample. We measure the human proteins, this is what it looks like. Okay, a variety of categories. I want you to focus on the green bars way in the left-hand side. These are the human immune proteins that are fairly consistent depending on all samples, right? Okay, what's going on here? So this human baby host had never seen a microbiota before. Here comes this onslaught of bacteria, and the human inflammation system said, wait, this looks dangerous to me. So the human inflammation proteins come screaming out of the background, trying to attack it. And eventually they'll kind of settle down into a symbiotic relationship. Okay, so there's not a lot changing in the human system, but we wanted to look, are there specific proteins that influence establishment dynamics? Pause for a minute. Okay, so we looked at a variety of infants here. Sorry, am I doing that or?
No, somebody, Nick Packin, please mute yourself.
Okay, Okay, my apologies if I'm doing any of that. So this is a little bit of a congested slide. I'm just going to show you the baby 19 and 21 we looked at before, and we marked across all of the details. You can see the little diamonds are when there's either sepsis or NEC occurring, and then the black bars are when the antibiotic got administered. And so there's a whole bunch of stuff that's a little bit hard to control. What I want to point out is every one of these babies got gentamicin at early stage, which is wild about, if you've thought about that, but why would you take a flamethrower to an infant that's developing a microbiome? And when asking these questions, nobody has an answer other than that's always what we do, because we feel like the babies are vulnerable at an early stage, but not, why would you give antibiotics if there was no reason to do it? Okay, anyway, we want to focus on this infant 61. This is a baby that had some interesting dynamics. The blue diamond is NEC. So this baby got NEC twice, necrotizing enterocolitis with blue bar, and then got antibiotics and recovered, and then got NEC again, and then got antibiotics and recovered. We thought this is kind of a cool test case. Let's take a look at what's going on in this particular infant. How is this microbiome being established and basically kind of, wobbling with time? So this is let me try to explain this curve. So on the x-axis is day of life in this infant from day 6 to day 82. And these bubbles are all microbial taxa. You can see them labeled by the first hand time. Let me walk you through this a bit. So we start at a particular point, and do you see how we have these waves of progression? Do you see how what looks to be fairly stable becomes sort of just waffled out, and it changes to something else, and then it changes as well. So there's these patterns that are going on that are very dynamic in here. And you can see, some of the ones, this is Enterococcus faecalis, a very common bacteria in infant that becomes very dominant, fairly early on. But if you look at this closely, you notice that some events happened here, right? So some traumatic things happened. What are they? One of them we know exactly about. That's an antibiotic administration. And so what happens in the microbiome, antibiotics are rough on microbiomes. So when antibiotics, and I think this is gentamicin, when this happened, you see the crash that occurred, but look at the rebound that occurred after this, right? If you look at the colors, you notice that this guy crashed and didn't come back, and this guy basically bloomed, right? So there's very specific responses that are coming here. And we usually want to give this talk, we all know this very well, right? You go to the doctor, you take antibiotic, what happens? You often get diarrhea, right? The doctor says, eat yogurt or do some other, put the good bacteria back in, right? You're basically trying to compensate for that a bit and stuff as well. One other thing confused us, you see this point right here? Something else happened before antibiotics that was a fairly dramatic shift in the microbiome. This early stuff is just wild oscillation, but this is a specific event that happened. And we didn't know that until we started looking at some of the samples. We thought, what in the world is going on there? So we wanted to, say, obviously there are things other than antibiotics that can push microbiomes around. So how do we explain some of this? So we went in, we looked at the proteins in the organisms. We noticed that some of the organisms had antibiotic resistance. Right? You can see this one, the Klebsiella and Enterobacteria cloceae had antibiotic resistant proteins. They're able to process that, but the faecalis did not, right? So when that trajectory occurs, that's exactly why the bloom is coming up in the red, and that's why the yellow crashed, right? It has to do with the protein inventory, the stuff that we're measuring. In the process of looking at that, we said, well, there's something else that's weird kind of happening. We're finding proteins related to copper resistance that are coming up at this point in time in the microbiota. What are these? And these are very specific. Two of these three key organisms had these copper resistant proteins, and one did not. And that exactly maps with what's happening here, right? So we looked at this a little bit more detail, and this postdoc chases down. It turns out the human host copper killing is very common. And so a lot of the human system will use copper-1 in the macrophages to basically kill pathogenic bacteria. It's a very common one, right? But some bacteria figure this out. So some bacteria have the proteins to basically say, I'm going to annihilate that effect. I'm going to take copper-1, and I'm going to use my protein machinery to turn it into copper-2, and that's less toxic. So the organism that can do that survived, and the organism that didn't were killed by the macrophages, right? So it's an interesting correlation. This is still a little bit cryptic, and there's a fair amount of stuff that we don't totally understand. But let me just pause for a minute. What's going on? When we look at these very detailed metaproteome information measurements, we got a lot of details about the proteins. We can start to chase pathways down, right? I showed you in that KEGG map, pathways related to antibiotic resistance or metal resistant are very specific things that we can kind of pull out. So it's still kind of a work in progress. There's a lot of details kind of, going on with respect to that. One question that comes up, when you take an antibiotic, does your microbiome ever come back to what it was before? And the question is not sure. Okay. For most adults, they're much more stable. Most of us, the microbiome is pretty established. If you don't overdo it with antibiotics, they'll rebound back pretty dramatically. Infants don't have that stability though, right? The pendulum's still swinging wildly. So these things are really highly perturbed. And then one of the microbiome researchers, his name is Marty Blazer at New York School of Medicine, is on this crusade about stop giving so much antibiotics to our kids. We're way over using antibiotics for kids. Kids come in with a little infection or ear infection or whatever and stuff, and instead of letting it ride it out, we're basically treating that, and then there's a correlation. Is there any wonder that obesity has gone up in our country in the last 20 years, maybe related to doing too much stuff for the kids? And he's written several books, Let Your Kids Eat Dirt and other stuff like that. You can take a look at that if you want. Okay, let me, I do want to save a little bit of time for discussion, and I'm trying to keep this way close. In some cases, what I haven't shown you, the mother was given antibiotics, right? When the mother goes in, mother may be having some problems with these preterm infants. What's your prediction? Do you think that antibiotic affected the infant? And the answer is yes. So that antibiotic is not invisible, even though the infant doesn't have a very healthy microbiome yet, there's perturbations that occur if the mother gets antibiotics. Okay, we've looked a little bit at this antibiotic resistance. Everybody knows antibiotics change your microbiome. The metal homeostasis was not really known. This was really kind of interesting that the human host can try to customize its microbiome, but these metal kills. And that was kind of an interesting finding. I don't know how widespread that is. We really try to follow longitudinal studies. So the most foolish thing, in my opinion, would be take everybody in this room and take one fecal sample and try to determine who's healthy and who's diseased. It's so dynamic, right? What you should do is take an individual and follow fecal samples over the course of several years and look at changes in the microbiome as a function of taking medication or health-related issues. A much smarter way to do it. Longitudinal studies are a way to kind of control for that. Hopefully, if even scared you with monozygotic twins, it can be different. And even when the genetics are held constant, that still can be a bit of an issue. So let me just kind of leave it at the last end that this field of metaproteomics, I'm very active in that. I'm actually a board member for an international group. I was in Norway about a month ago. We meet in Europe about every 15 months. And we're basically trying to propel this field forward. How do we do a better job measuring? And there's, about a handful of us in the world that are basically trying to push the technology better, because I think it's going to be really fruitful. I think it's an incredible way to reveal information about a system that's still fairly poorly understood. Let me just kind of end up with a few acknowledgments then. Jill Banfield is a longtime collaborator at UC Berkeley. I did the acid mine drainage, that pink snot on the pond with her, got a variety of other microbiome stuff. Michael Morowitz is a pediatric surgeon at Pittsburgh that gave all the baby samples. I feel like a new father. I've got freezers full of baby poop, little vials of baby poop. And then some very talented graduate students and postdocs that have worked in the group that have been incredible sources of information. And these people have so much creativity. Give them an opportunity and they'll just run with it. So here's a picture of the group. I want to point out a couple of people. Samantha Peters did a lot of the work I've talked about today. Matthew is my 31st PhD a month ago. So he and Manessa have left now as of December. I read 535 pages of dissertation in the last couple months. So thank you for your time and interest, and I'd be happy to chat or have some questions if you have.
What would be a good cocktail to drink?
Yeah, that's that's an interesting. I'm actually working with a company in Maryland, and the guy's coming down tomorrow. That's basically trying to customize the microbiome. And he said, Bob, you're one of the few groups in the world that can measure these really deep. If we take a bunch of your information, can we come up with cocktails to basically try to customize the microbiome? Now, let me try to give a shorter answer because they can get real windy about this. Are you familiar with the fecal microtransplants, okay, which have some horror stories? It's worked really well, and it's also been really bad. So getting a microbiome altered is a dangerous process because we don't fully understand what we're doing quite yet. And I would contend if you go down to Walgreens, you can buy probiotics, right? You can buy anything, but most of them are the wrong stuff. They're lactobacillus. Most of them have too much of that anyway. The good thing about them, they're not really bad for you. You can buy them and you're just going to make healthy toilets basically because the stuff's going to run right through, right? But what you're wanting to do is customize stuff. You want a smart bomb. You want to basically say, I'm missing this member or this function. I want to go in and seed that. And it's not quite possible yet, but my prediction is that within 3 to 5 years, we will have customized probiotic stuff. And I think that all the way from obesity to other stuff, I think that we could even treat some diseases where we're trying to throw antibiotics at it and we could do it in a different way. So some of this stuff is starting to be called precision medicine in a way because it's correcting stuff in there. So that's a windy, soft answer to your question. What about the yogurt? Yeah, I mean, yogurt is great. I mean, that has beneficial effects. All of those are kind of generic though, right? I mean, they're sort of effective, but they're maybe not really what you need. You could be very different from your neighbor. So if your doctor understood that a bit, but let me give you a practical example. I mean, you have a heart problem, you take medicine, right? You're throwing a pill down your throat. When the pill gets in your system, can I show you the bacteria? They're going to touch it 1,000,000 times before your system gets it. So the bacteria are going to try to degrade that and metabolize that pill as well as the human system. So in my mind, it's a very naive comment. I'm not surprised that medicines work different in different people, partly because this microbiome is not playing an insignificant role in that. There's all sorts of crazy stuff. Like, for example, that you and I don't have the enzymes to degrade plant polysaccharides. So if you didn't have bacteria in your gut, eating salads would be meaningless, right? You'd have terrible indigestion. So the bacteria have the enzymes that are degrading the plant polysaccharides for us. So this whole symbiotic relationship is really kind of critically important. Diversity matters. I want to come back to your point. Most of us that are healthy, whatever that means, have billions of types of bacteria in your gut. And what happens in every disease state, whether it's diabetes or Crohn's disease, whatever, this bacteria diversity collapses. And what was really rich complexity becomes very shallow. And you can imagine that's not a good thing because if you have 500 members and you kill one of them off, somebody else takes the job. But if you only have 100 members and a critical one gets killed off, you're in trouble, right? This is called shoaling. It's a word actually in the micro. Shoaling means whatever disease state, this complexity collapses, and that's not a good thing. You're less resilient to doing stuff.
Any data on how the GLP-1 drugs affect the microbiome?
Not that I'm aware of, actually. I think that that's a little, early on. Now, some of the medical profession is a little bit cynical about, you guys are trying to relate everything to the microbiome, and that not everything can be there. There's still the human system that's important, and they're related together. So we have to be careful we don't swing the pendulum too far in the other. I would also argue physicians have to be careful that they don't ignore the microbiome, which I think we've done for far too long and stuff.
Are there any medical facilities in East Tennessee who actually test the microbiome and give you some advice on?
Not that I'm aware of. I mean, there's places you can send a fecal sample and you can, have some surveys done or whatever and stuff. But, I picture some of this stuff to be sort of 2 to 5 years out from practical implementation. You can tell some of the stuff I'm talking about is kind of fundamental. You can see holes in it where we have incomplete pictures of information. But I think it's kind of exciting to march down that process a bit and stuff. Huh? Frank, did you have a question? Okay.
I believe you told us that there are about 10 to the 14th live bacteria in each of them. And I take a 5 milligram or a 50 milligram pill that has profound effects on my heart behavior, which means that there are very, very few molecules in the pill per bacterium. How does this play out? Does it mean that the pill is extremely specific, or what's the...
It's a great question. I'm afraid I don't have much of an answer for that because all of that's a bit complicated. There's certainly pills that have ingredients that bacteria won't care about or can't do anything about, right? And then there's other stuff that they can partially metabolize, and I think it's all over the map. It depends on kind of who's there and stuff. So I don't have a lot of information. I've taken a probiotic daily for probably a decade, and it's, I feel like it can't hurt, right, and stuff, but let's go way in the back there.
Just a couple of comments to hear what you have to say about it. This was in probably The Economist about 2 years ago. I think it was World Health Organization that solicited some specialists to look at microbiome. They were looking at monozygotic twins, ones that had been malnourished. The goal was to see if the malnourishment regimen that's used around the world could be improved. Nobody had improved it for decades. But they looked at some monozygotic twins and found that had previously been malnourished, and some now, later, one was doing much better than the other one. And they looked at their microbiomes, found them to be very different. They looked at like 16 different diets, and they had come up with about 6 that showed a dramatic improvement. And again, it was not just weight gain. They were measuring 20 plus different blood proteins that were proxies for different health measures. We found that once they got the microbiome similar, that even if the calories weren't as much, the child would gain more and all these health parameters would be better. So I'm wondering about if the complexity is maybe not so huge. The ones that were best had just 6 things in them. I mean, in terms of the diet, it was bananas, and it was rice, and it was soy, and it was chickpeas, just a few things, and peanuts. But also the yogurt issue, when they look at observational studies, I mean, nurses studies, been going on since the 70s. 17% all-cause mortality reduction of people who are eating yogurt, even just a few times a week. So maybe it's really complex, but people aren't living in a complex world where their diets are made up of thousands and thousands of things. Maybe it's easier to have a healthy microbiome with doing a few basic, simple things. Is that?
Yeah, there's a whole discussion on that. I mean, you're exactly right. We're trying to draw correlations from things we fairly poorly understand, and that's very dangerous. And scientifically, you got to be careful about doing that. And, we know parts of information. I have a couple of comments I want to make. There's a tribe in the Amazon that have probably the healthiest microbiome on Earth. And they wear relatively little clothes. They're in contact with dirt and everything around. They have no preservatives in their food. Everything's fresh. They have no eczema. They have no skin. There's a lot of things even related to microbiome, whatever. We in the US are doing a really bad thing. And you know what? And we have way too many fast food restaurants. We eat way too much sugar and fat. The microbiome does not like sugar and fat. It's going to modify if you're not eating as much plant material. What's happening in the US by and large is we're shallowing. All healthy US people have shallow microbiome relative to what it used to be. In fact, let me pull off on a tangent. There's actually a vault in Sweden that's a repository for microbiome. Some people know about that, right? So some scientists have figured out years from now, we maybe regret the fact that we may want some seeds to go back. So they're basically putting some of these stocks in there. I want to come back to the obesity thing a bit. Marty Blazer has a couple maps I've seen on talk, and he's got a colored map of the US where he's colored by antibiotic abuse. And it's terrible in the Southeast where we live and whatever. Then he lays a map of obesity on top of it. And it's almost a 1-to-1 correlation. It's frighteningly correlated, whatever that means. But let me come back to the obesity thing. So I've also done some work with Jeff Gordon at Washington University. And if you've read much, Jeff will probably win a Nobel Prize at some, not the race car driver, the other guy. Jeff Gordon will probably win the Nobel Prize because he's one of the leaders in microbiome. He's done a lot of stuff with notobiotic mice where you can knock out their immune system and you can seed them with the humanized microbiome, and you can watch that occur. And he's done a lot of diet oscillations where he's looked at the microbiome from lean mice and obese mice. And so what's your prediction if you take the microbiome out of an obese mouse and you put in a lean mouse, what happens? The lean mouse gets fat. It predictably exactly without, and so that microbiome is controlling a lot. So your point about it's not even the calories as much. So Jeff is on a crusade right now to basically try to work on malnutrition in Bangladesh. It's basically this concept, but can you correct microbiome of, young children by adjusting it in a way to basically make them absorb better? It's not just about the lack of food, it's the microbiome screwed up. I don't, he's been working on that. I think the Gates Foundation's funding him or whatever. So.
You sort of gave us a little bit of a conflicting direction, so I'll check this out. You mentioned that the probiotics don't have anything useful in them, but then you admitted that you took them every day.
Yeah.
So I guess the correlation in that is what, with current probiotics, what's our understanding of what's in them and perhaps how useful they might be?
Yeah, you got to be careful there because even my information is a bit anecdotal. I don't think they're not bad for you. I just think that they're not customized and they're too generic kind of across the board. The older ones used to have a lot of lactobacillus, and that was probably the least effective. The newer ones have it's called rhamnose. I think there's a particular strain of bacteria that are much more preferential. So I think I take them because I think they're a good thing to do, and I don't know whether it makes a difference or not. What I'm excited about are the customized ones, where I'd like to know what I'm missing relative to a particular body type or disease condition, whatever. And so I have two daughters, by the way. My 40-year-old daughter has Crohn's disease. My 36-year-old daughter doesn't. I'd like to, I should have taken a fecal sample from each of them and just kind of seen what happened or whatever and stuff. Rich.
So you've mentioned cultural differences affecting microbiome.
Yeah.
Health effects on it. What about, can you do your kind of analysis on archaeological samples and look at sort of the evolution of the microbiome along with human evolution?
That's a good point, and that's come up a few times before. The stability of those samples are a bit of an issue because most of the proteins are gone and you can't reach back very far. But that's why going to other places that are a little bit more untouched, like the Amazon rainforest versus the Western world, or whatever and stuff like that, and you can there's a correlation there. So it'd be interesting to look at that and see what people have monitored changes over the past, I would say 20 or 30 years at least, and there's observable changes even there, especially in the westernized countries.
Yes, did Jeff Gordon do the reverse by implanting the lean microbiome in the obese mice? And what, if any, result?
Okay, do you hear his question or do the opposite? What if you, so I told you if you take the microbiome from an obese mouse and put it in a lean mouse, the lean mouse gets fat. He's asking about the opposite. If you take a microbiome from the lean mouse and put in an obese mouse, what happens? Nothing happens. An obese mouse within the time frame that's been observed, that mouse doesn't suddenly get lean. So there's some things that either have a longer time period or you started to go down a trajectory that is a little bit, maybe the lean is the healthy and the obese is the disease state. And as long as you're lean, you can go to disease. But once you're diseased, it's hard to swing the pendulum back a little bit and stuff, right?
It's my understanding Crohn's disease is strongly correlated with hereditary. And so then how does that relate to that biota? And what you said is babies, when they start out like not much there.
Yeah.
So do they contact it subsequently from their parents, or how's this go?
Okay, yeah, good point. And the genetic basis of Crohn's disease is only about 15%. It's fairly low. And in my particular daughter's case, as far as we know, there was no basis. But let me give a quick story for my daughter. I've got a theory about that. So my wife is pregnant. I'm in graduate school at Purdue, and my wife's water breaks at home. So we rushed to the hospital. And my daughter Rachel's not born for about another 20 hours after that. And on the way out, I think my daughter took a breath or something like that. She got a blood infection very early on. She went to neonatal, about died, and she was really very critical condition. So the doctors came in, what do we do? So they said, we're going to try antibiotics because she's got an infection. The first set didn't work. So what do? You up the ante, right? So they start bringing out the flamethrowers or whatever, and they basically got to the point where they stabilized her and she survived. It was great and stuff. And then Crohn's disease, we were kind of talking before, Crohn's disease typically develops in usually late teens to early 30s. That's very typical, right? In the 20s is very typical. My daughter got Crohn's at age 8. We took her to Children's Hospital at UT, and the doctor was like, I rarely see this in a kid at that age. My theory is we screwed up her microbiome. That trajectory got pushed off. Now, we saved her life, I mean, fairness or whatever, but on the other hand, there's no genetic basis as far as we could tell. And my other daughter's eating the same food and doesn't develop it. So, still a subtle thing. There's not one marker. It's a complex signature. We can go back, yeah. Yeah.
What effect do anti-inflammatory drugs have on the gut biome? It causes disturbance.
Yeah.
Do they actually alter it?
Yeah, I don't think I can answer that. I think that there's not anti-inflammatory drugs. Yeah, they may have some beneficial effect, but they may be highly individualized too. I mean, I think we don't know the range of what that would look like. Let me go way back here.
My son got Crohn's at 4.5.
Oh,
And for 2 years he was in India. Then for 2.5 years he was here. And he's 40, the same another 39. The culture had something to do at the time of birth. What happened?
a very good point about it. That's why, these are complex signatures and, there's ways to perturb them, but they're not always clear about how, or certain individuals are more vulnerable than others, right? There's other factors at play. It could be genetics, it could be other stuff where their microbiomes might be more volatile than their sibling or whatever and stuff, right? And I think we'll learn some of that, but I don't think we know that yet.
I had a mass spectrometry question.
Yeah, please.
Yeah, 20 years ago, there was a competition, how big a molecule could you analyze with mass spectrometry? And how much has that improved and what are your limits now?
Yeah.
On how large, you can't put the whole bug through the mass
No.
spectrometry.
Yeah. Ray asked a good question. We worked together some years ago, right? Yeah. So yeah, I didn't really talk about the mass spectrometry, and it's kind of interesting. There's a whole, that whole field develops, and it's all underlying part of this. What's really Ray's question about how big of a protein or molecule you put in there, and the answer is electrospray, which won the Nobel Prize back several years ago, is a new type of ionization, makes multiply charged ions. So all of a sudden you can take really big proteins that we never could fly, and you can actually fly an intact virus. You can fly protein complexes. So you can do really big protein molecules are possible. For most of our stuff, it's called bottom up. We basically throw a grenade at it and, rip the pieces apart. Kind of like taking your car to the shop and the mechanic puts every piece in the driveway and then tries to find out what's wrong. That's kind of what we're doing. So we don't, but the instruments have become quite a bit more sensitive. They're much more accurate and they can lift big molecules. So it's an unprecedented. I have a graduate student just starting with me, and I walked in the lab the other day and I told her, do you realize how phenomenal that is what you're doing? I would have loved to do that, 10 years ago, but the instruments are just getting better and stuff.
So what is the typical size of a molecule? 10 billion?
No,
100?
the proteins, most bacteria proteins are about 25,000 Daltons. 25 kilodaltons. So most proteins are fairly small to moderate, depends on, some of them are part of complexes, so they're not incredibly large. Let's go way in the back there.
Is there a correlation? Is it just what you see in microbiome? Can it be psychological stress?
Yeah.
Trauma, how much physical exercise somebody gets?
Of course, there's a lot of details. Well, let me come back briefly to the probiotic. There's, just simply throwing bacteria in your gut is probably not real effective for all the things we've talked about. So people are trying to put prebiotics with probiotics. So in other words, it's the food source for the bacteria with the bacteria. And I think they're called symbiotics. You put them together. When you give a favorable bacteria, give it a little kickstart by a nutrition that it needs or whatever and stuff. Excuse me, but you're exactly right. I mean, there's other physical factors in our system that play on this, right? So the microbiome can't develop totally independent of this human system that has, inflammation markers and all sorts of other stuff or whatever that, So, yeah, let's go up here.
These complex systems you look at, they seem more daunting than chemistry must have been before the periodic table.
Well, that's true. That's really, I mean, some people say, why are you even doing this? You're missing a lot of this stuff. And my point is, yeah, but we're measuring some of this stuff, right? Sometimes you got to peer through the dark curtain and or open the doors a bit and start to learn. Are we naive? Are we missing? Absolutely. But we knew nothing before, right? And so we're starting to bridge forward a little bit or whatever.
So my question, kind of following on that is, when you try to get a handle, you mentioned probiotics several times. And is there any problem with talking about probiotics, only those that will enter through the one end of the tube? One of introduce them in the other end?
Yeah. I mean, yes, there's a whole range of stuff. I went to a meeting in Boston that was a commercial companies that were probiotics, and they were talking about formulation systems. You know, when you take a probiotic by mouth, the stomach's going to kill most of the bacteria. So you do not want the pill to release in your stomach, right? Because the acid's going to rip it apart. So most of the pills are coded that they don't release until basic pH, which is in your intestine. You know, you can certainly go in the other direction, which, okay, and that's the fecal, stuff. Now fecal transplants are kind of disgusting. Even nurses and doctors don't like to do them. You can imagine why. So there's several companies trying to figure out, back to your question about, are there 20 or 30 favorable bacteria that we should put in a cocktail that's a colorless liquid, and we make a fecal capsule out of that rather than fecal material. If we can capture the function in 20 or 50 bacteria rather than 2 billion, we can get the same effect, right? And stuff. So it's kind of exciting. There's a lot of work going on in that right now. And again, don't know what's developing. I'm quite interested in doing it. And the guy that's coming tomorrow, we're going to try to figure out if there are ways to customize the microbiome. Can you go to a microbiome and say, I want to kill that function, either that bacteria or that process with an antibody or something like that, and I want to dial in what I want. And it's a little bit of a pipe dream right now, but we got to start the process, would be my opinion. By the way, I'm having a great time here. I don't think anybody's going to yell at me for another hour to get back to work, so I'm happy to.
You already said that because of fecal samples, that implies that a lot of this bacteria moves out of your body. So the corollary question is, what's the turnover?
Yeah.
I mean, this stuff's coming out, it's got to either grow or come in again. So what's the, does it all turn over in 30 days or what's the?
Yeah, that's a good point about that. The fecal sample is kind of a time average, what came all the way through the small intestine, the colon, right? And so, we want to capture that right away, though. I mean, fecal sample, we got to grab that right away and freeze it so the protein signatures don't change. So I've got to be a little bit careful about that. I actually don't know the answer to that. It depends on the type of bacteria. Some of them are really attached strongly and they turn over very slowly, and other ones kind of get washed through a little bit, better. Now, the one thing, let me go back to the probiotics. We're dealing with that for a little bit, but in your microbiome is this whole, community of, that's sitting on your epithelial cells, and they're all established. They're biofilms that are basically grabbed on. So now you throw a pill in with something you want to seed. Well, there's no room for it to land, right? Or the members that are already there are going to kill it well before it ever gets a foothold. So even though it's something you want, you just put it in the toilet because it can't, So, people are looking at ways to establishment is a big issue there, right? And we kind of know some of the members, but we can't get them stuck, in ways of what. Carolyn.
Is there a relationship between the microbiome and inflammation? Do some microbes make it worse, some make it better?
Yep.
Or how does that work?
Carolyn's question about inflammation, exactly right. Some are really bad actors when it comes to stuff. And you read, in the news, Clostridium difficile. Does that ring a bell? That's the bad one for Crohn's disease. That's killed several people. You realize about 3/4 of the people in the room have C. diff in your gut right now? And the reason it's not a problem is the high 500-pound gorillas, the Bacteroides longum and dorii, are keeping it at bay. And they're more dominant. They're basically saying, you can exist, but your function is very controlled. It's like this little township or whatever. But if something gets killed off, if a 500-pound gorilla gets killed off, that's an opportunistic pathogen that takes a hold and away we go and stuff. So some of them are really clearly markers of inflammation. Sometimes in ways that we don't totally understand and stuff.
You could say another word about that translocation process where bacteria or bacterial products migrate through the abdominal wall because of cross integrity.
Mm-hmm.
The they have seen correlations that are probably causative of, impaired glucose tolerance.
yeah.
And promotion of, translocation, which incites all these inflammatory processes. So the diet somebody eats can be tied to inflammatory bowel disease. And even when you look at something like COVID-19, it's not the virus as much for all the long COVID processes.
Yeah.
It is the massive unregulated inflammatory response that has created many of the problems. So you know more about the translocation.
That's a very good point. I mean, you bring up a good point too. A lot of times when we think about stuff, and I got to be real careful because I'm not an MD and I'm making comments and suggestions, which are, you know, hopefully not crossing the line. But we oftentimes think of systems as linear. You think of the human system or the genetic system or the microbiome, and we compartmentalize because that's where our brains work. But the problem is they're not. They're spider webs, right? Everything is tied to everything else. When you rattle one part of the cage, everything else adjusts a bit. So inflammation and human genetics and the microbiome and the population and a trajectory problem you had a year ago, those all factor into what's going on right now, right? And stuff. So, you know, you took antibiotics 5 years ago and your microbiome didn't totally rebound to the same thing. Now you get COVID. I mean, yeah, it's kind of open-ended about, but it's pretty clear that we should study it in more of a context, which is part of my research is systems biology, which I really love, is the standpoint about we can't be too myopic. We got to step back and we got to kind of start to look at this system as a whole because that's the way it operates, right? But,
What's the role of fungus?
yeah, that's it. That's a good part. Yeah. If you read any of the WHO reports, the Candida albicans, and there's some other stuff that are frighteningly increasing for a variety of things. So, you should have nailed me a little bit earlier. I talked way too much about bacteria. There's not so much archaea in the gut. There's some methanogens, related. There, other ones. But the fungi, for most healthy people, there's not much fungi in your system. There are disease cases where they really get a foothold and they can get really nasty and they're really kind of increasing and stuff as well. So for the most part, a lot of the pathogenic fungi are not real happy at the body temperatures, at least right now. And so, yeah, that could change a bit and stuff as well. The other compound that I didn't mention, I would just want to briefly bring this up. We talked about where are the bacteria at, and your epithelial cells, the bacteria have to be very spatially located. If they get too close, the machine guns will kill them, the immune system. If they get too far away, they get flushed out. So there's there's this, and people have wondered what keeps those at bay? What's establishing that zone? It looks like it's viruses. Viruses are loaded in that region, and the virus predation, if a bacteria gets too close, the virus will kill it in the region before the human system even does. So now we're back to the system. This is all balanced together in a very beautiful way in my mind. Virus, microeukaryotes, they all play a role in this. I mean, I'm not sure fungi don't even have a healthy benefit under some cases, whatever.
So do we have viruses, a large number of viruses that float around all the time?
Absolutely. Yeah, there's probably 10 times as many viruses as there are bacteria.
Oh, really?
So when you think about that, in fact, I learned the other day, I mean, there's a lot of work on the ocean microbiome, and the viruses in the ocean will kill, let me see if I get this right, 2/3 of the 40% of the bacteria in the ocean are killed every day by viruses. 40% of the billions of bacteria, because of virus predation is just, and it's part of the control system, right? But so. Thank you very much.